So welcome everyone to episode 10 of our RSDP Connections webinar series. My name is Rose Clark, and I'm a Conservation Corps volunteer serving an AmeriCorps term with the Regional Sustainable Development Partnership of the University of Minnesota Extension. My role is to help to connect the natural resource work groups across the state and all of the greater Minnesota regions where we support community-driven sustainability projects. And as part of that, we began this webinar series to kind of help our boards and work groups to learn more about the University of Minnesota, the resources that it has, and the opportunities for collaboration, and then also to share a little bit between the regions about what's happening. So today, in light of that, we have with us Matteo Convertino. He is a U of M assistant professor and engineer in the Department of Public, or School of Public Health, Department of Environmental Health Sciences. And he's going to be sharing a little bit with us about his research and some about his work. And then we're also going to be sharing some information about our RSDP Statewide Coordinating Committee meetings because in 2 weeks we'll be having one of our meetings, quarterly meeting, on Thursday, November 3rd, and the topic is natural resources. So we're very excited to have this opportunity to kind of build on our natural resource work. And yeah, so you'll be hearing more about that later. But right now I'm going to turn it over to Matteo to tell us a little bit more about what he's doing. So pass the ball to you. You can go ahead. Oh, sorry, a couple of other things I forgot. If you do have a question during the webinar, excuse me, go ahead and go to the chat box on the right-hand side of your screen, type a question in, and I'll read it aloud to the presenters. And at the very end of the session, I'll have a poll that I'll run. So if you could take that for me, it helps me with my Conservation Corps position. So with that, now I will turn it over to you, Matteo, and you can go ahead and share with us your presentation.
Okay, perfect. Well, thank you very much for inviting me. As Rose said, I am an assistant professor in the School of Public Health and Engineering at the University of Minnesota. My background is in civil environmental engineering, although during my PhD I expanded toward an area that is called biocomplexity engineering that tries to simplify the complexity of the world and translate that into mathematical and computer models that try to address problems that the population faces, from ecological issues to health issues. And so my role in the School of Public Health is really trying to develop those models to try to control not just a disease issue, but also issues related to environmental exposure that can affect both the animal and human population. And the topic that I'm going to describe today a little bit is actually a project, one of the projects that I have with the Grand Portage Reservation and a variety of different colleagues here at the University of Minnesota in the College of Veterinary Medicine and outside the university at the EPA and the Minnesota Pollution Control Agency about the Grand Portage Reservation itself. So the problem, we started this collaboration because even in Grand Portage, that seems like a very pristine and untouched area, is actually subjected to a variety of different issues, starting from overload of nutrients, presence of pesticides, spills, and other micropollutants that are actually causing ecosystem impact. And this ecosystem impact does not just affect the species that live there, but also they can potentially affect the human population, because these people over there, they actually live by consuming subsistence food, and that subsistence food are basically the species that are affected by these micropollutants. And so there is a variety of complex phenomena that needs to be understood, and trying to, for example, quantify food advisory consumption that are trying to decrease the exposure of people to some micropollutants that are found in this in this subsistence food, and at the same time looking at the ecological response overall. So what they're trying to do is really trying to link the observed pattern, for example, pattern of concentration of micropollutants or pattern of animal death or contaminated species to the processes, because we don't really know, for example, where these micropollutants are coming from, how we can control them. And so we are really trying to link together these two things that are not really well understood, and traditionally they're not linked together. If you think about like the field of toxicology, epidemiology, they typically don't really work together. So technically speaking, toxicology, and even if you think about ecology, they're always trying to understand the processes behind the pattern that we observe. In epidemiology, instead, the attention is really to try to characterize pattern in population without really digging deep into the basic biology or the basic mechanism. So in this project, we're really trying to do both. And we adopt this One Health framework in which we model simultaneously the environment, the animal population, and the human population. And so the type of data that we have, for example, are like data that are looking like this. So for example, we have the mercury levels in a variety of different water bodies. And mercury is just an example. We have, I think, more than 55 micropollutants, and within these pollutants, we also have heavy metals. And as you can see, what we notice is actually that the variability of these micropollutants over time is increasing, and overall, also the magnitude of these micropollutants is increasing. What we want to understand is what is actually causing this variability, and what are the environmental pathways that are responsible for this variability over space and over time. Now, just to give you an idea, since we are looking also into the ecological effect, if you consider the fish biomass itself, and fish is just the fish population is just one population that we're considering among many others. But if you just consider the fish biomass, you can see that for any lake, including obviously the Lake Superiors, we started to have a decline starting in 1986 that is not really understood, in my opinion, at least looking into the literature. And so obviously this is not just an ecological issue, but it's also a human issue because first of all, we have a decrease in abundance, which obviously affect the fishing community, but at the same time, there are many communities that actually survive on this fish population, and they may get contaminated, and not by chance, actually. We observe, for instance, that the total number of cancer cases in some population is actually increasing, not really exponentially, but with the positive trend starting from 1986 as well. So there are several issues of concern. Now here, I just listed actually the percentage of lakes with the detection of the micropollutants that I was talking about. Now, I'm not really an expert in these micropollutants. We are working with experts that actually know how these micropollutants behave in the environment. My goal is really to try to model, embed into a computer models that dynamic in order to understand what are the potential sources, what are the potential pathways, and how animal and humans may be affected. And now that at the end may translate into potential health outcomes for the population that we consider. Now, This one was actually a survey in 2012 for all the lakes in Minnesota. What we did is actually a preliminary field detection for some water bodies in the Grand Portage Reservation. And we found a smaller subset of micropollutants, but still these substances are actually quite worrisome if you consider their potential health outcome, because they are, some of them are actually endocrine disruptors that actually can affect the human body quite heavily. And so we're trying to understand what's the level of concentration of these substances and what is the association between the fishing places and these substances. Now, what we proposed actually to the LCCMR program of the state of Minnesota, that is actually funding us starting from next August, is actually a 2-year study considering actually a larger geographical area, and this area is actually called the 1854 seeded area, that is much larger than the Grand Portage Reservation itself. And we are essentially having 3 components. One is actually environmental sampling that is performed by the people in the Grand Portage Reservation. They have an environmental laboratory that is actually taking care of all these things. The sampling of subsistence species is actually performed by the College of Veterinary Medicine at the University of Minnesota. And on my side, I'm doing also all the computer stuff. So I'm basically taking all the data that they're sending me, considering the environmental information, for example, elevation, river network, and climate, and trying to model the pathways that are potentially responsible for the issue that we observe. So essentially, there is this 3-step that is starting from the chemical detection, analysis of the biological effect in subsistence species, analysis and modeling of ecological effect, and the potential forecast for population health outcome for the region considered. Now, how all this project started? This project started actually from a population signature program sponsored by the Institute of the Environment, and we grouped together all the people that I mentioned, so people from the Grand Portage Reservation. Actually, at the beginning, there was also one scientist from the University of Saskatchewan that is not actually part of the group anymore, but we were considering him because there are many areas in Canada that are actually very similar, hydrologically speaking, to the Grand Portage Reservation, and they're observing the same issues. So eventually, in the future, we are going to link with them and trying to compare and contrast our funding with their own funding. And after, as I mentioned, there is the Minnesota Pollution Control Agency, the College of Veterinary Medicine, and actually USDA was another partner at the beginning because we were trying to source some funding considering their own food safety program, since this one is actually a food safety issue. And this is actually a picture of all of us after the first meeting that was actually in Grand Portage itself. As I mentioned, there are actually other species that we're considering. Some of them are subsistence species, so they're used for consumption by the population, and some other are just indicator species. So they're not necessarily subsistence species used for consumption, but we wanted to track them because they can be potentially affected by the same micropollutants, just because these species use the environment as we do, and then they can be exposed and they can actually absorb these contaminants, and they can have actually a quite similar response function of the human body. Now, to be a little bit more detailed about what I'm personally doing, I would like to spend some time, maybe at least 10 minutes, talking about it, because I think it's really relevant. I think that mathematical models are tools that should be used in public health because they are tools that are able, first of all, to bring together all the data and try to understand the relationship behind the data that we collect. This one is important because we always want to understand what are the relationship of the variables that we measure. And secondly, when we understand that, we want to forecast what's the likely situation at the present time and in the future, considering, for example, different management options. And this is really precisely what we want to do in this project. So what we are characterizing is actually each area that can be defined as a community in terms of their environmental feature. That can be, for example, the drainage area, so the total amount of water coming from a particular place, the level of concentration of all these micropollutants, and how these micropollutants are actually transported downstream. So the core of the model is actually an hydrological model, which is actually refined by some consideration of the landscape feature in terms of vegetation that can actually affect the transport of micropollutants. Now, when that is actually mapped and the current state is predicted, we can actually forecast some potential health risk using some exposure response function that actually have been studied in the past. Now, without going too much into the details, essentially the modeling steps are basically the following. We receive, for instance, some information about cases or some information about concentration of micropollutants. Here, a case is defined, for example, a species or a human is actually found with the level of concentration that is higher than the normal, the threshold that is established. Starting from that, we can couple to essentially the environmental model in which we have, for example, the river network that is responsible for the propagation of micropollutants. We have the forcing that can be, for example, the precipitation pattern that is actually essentially informing us how much water we have in a particular community. And after we include a probabilistic element that explains all the variance of, let's say, cases or concentration that is not explained by these environmental factors, because we actually we cannot explain all the time the variability of certain micropollutants. There is a certain unexplained variability that may be related to other factors that we don't know yet. And all that comes into this predictive model that tries to identify the optimal management strategies constrained to resources that can be, let's say, budget available, or stakeholder mental models, where stakeholder mental models really means what people want to do, what people prefer to do. So there are certain areas, for example, that are designated for fishing, or some areas that are somewhat used by the local population for hunting certain species. So obviously, the optimal management solution needs to consider the stakeholders that are the primary users of the landscape, and then that is actually considered into the exploration of the optimal management solution. Let me see what I can do here. This one is just essentially a multi-resolution visualization of the study site. This one is actually just focused on ground portage. So as I said, the area that we are trying to explore is much bigger. But here I made this visualization because I wanted to point out that the knowledge and the mapping of the environmental metrics, for example, the river network, is crucial for understanding the propagation of these chemicals into the landscape. Now, what are the preliminary findings that we find? The preliminary findings that we find are the following. If you consider the total concentration of all micropollutants, and this one is actually in a log scale. So as you can see, here we span at least 3 or 4 order of magnitude. Okay. of this integrated concentration of micropollutants. And you plot that as a function of the drainage area, you can see that you have this trend where starting from the small river to the biggest river, first the variance is actually increasing. So essentially, the more you go downstream, the more you observe a higher variability. But secondly, what you observe is actually somewhat a negative trend. Now, this negative trend is actually kind of spurious because as I'm going to show you in the next slide, if you actually remove the total drainage density of the Lake Superior, which is actually way too big with respect to the area that we consider, you actually observe a positive trend. And again, this one is in the log-log scale. So this one means that you have a power law increase of the concentration of micropollutants as a function of the drainage area. And again, this one is worrisome because essentially you have an accumulation of micropollutants going from upstream to downstream, which grows very, very fast. This one is actually growth that is much, much higher, much faster than an exponential growth. And if you look at each single micropollutant, you have first that magnesium is actually the one with the highest concentration. The second one is selenium. Third one is lead. Fourth one is mercury. Fifth one is actually arsenic. So all of them are quite worrisome, both for the ecological receptor and the human population that we consider. Let me say again, the first graph was actually considering the total concentration in whole the Lake Superior. And then the drainage area over here is basically the drainage area of the whole lake. If you remove that, and that's actually wise to do because we are actually interested just into the coastal area that is essentially following the north shore of Minnesota. That's really our focus because most of the population is actually living on those subsistence species that are along the coast of the lake, not really the whole lake. And so this one is just this decay is actually just an artifact of the fact that we consider the whole lake. Let me skip this for a second and let me show you this one. Now, if you consider, for example, mercury, and you try to get the sample of the fishes that are actually consumed, you can see a relationship between the variation of the concentration of mercury and the variation of fish length that is, again, following this power law distribution. So basically, this means that the concentration varies, grows more than exponentially with the fish length. So in a sense, the larger the fish, the higher the accumulation of mercury. But that's not that trivial because if you observe different species, actually they bioaccumulate this micropollutant very differently because all these fish population, they follow a very different power law distribution. So here I'm actually just showing the average power law distribution, and we have some fishes that bioaccumulate much more than normal, and some others that bioaccumulate less. And also, if you notice, you have that for the same species considered, you have a difference in bioaccumulation according to the water body where that fish population is placed. So for example, if you consider brook trout, you have that the brook trout is actually bioaccumulating more in the swamp lake rather than in the Taylor Lake. And so there is this spatial heterogeneity that needs to be understood because it is important to, first and foremost, it's important to make this fish advisory communication dependent on the place where actually the people fish. It shouldn't be like an homogeneous fish advisory because different places carry a risk that is very different even for the same fish population. And this one is just showing a synthesis of what I just said. We have abnormal bioaccumulation in some areas and lower bioaccumulation, which is actually a safe level for some other areas. And most of these areas are actually the head stream of the river basin that we consider. Some of them, for example, in this case, this yellow perch is actually bioaccumulating at levels that are much higher than normal, and for the same lake, that is actually shared by other species. So actually, this trout lake is actually a pretty pristine lake, but despite that, we observe that the yellow perch is actually bioaccumulating a lot. And again, you need to think that these fishes move. So maybe this level of mercury is not related to the trout lake, but it's related to another water body where these species are actually coming from, and they carry the level of contaminants. So the association between fish presence, micropollutant presence, and contamination of water is not that trivial and needs to be mapped carefully. These are just other examples in which you see that for the same water bodies, and in this case are actually the coastal areas of Lake Superior, you have a bioaccumulation that is different for different species. And that's basically, these are just other examples. What's interesting is that if we analyze this data actually across the whole here, we can see that we have a variability for the months that we consider. So we find actually peak in micropollutant concentration during the summer, starting from, let's say, June, mid of June, and to the beginning of September, we have a quite large window where actually these micropollutants are reach the highest level. And this one is somewhat worrisome because that's the same season in which obviously most of the people fish. And then even this one is actually extremely important for informing the people. And this seasonality is clearly manifesting the fact that these micropollutants are somehow more movable after the winter season, and so they started to actually propagate through the landscape at much higher rate than during the winter. Let me see. All right. So I think this one is one of the last things to talk about before I conclude. One of the most interesting things, as I mentioned before, is try to map the relationship between the water bodies that we have, considering their potential likelihood to actually be subjected to the same micropollutants that is actually coming from the same source. So by considering, for example, the variability of mercury in different places and analyzing how that variability changes over time, we can actually map the network of water bodies and the directionality of transport that is actually informing us how that micropollutant is moving through the landscape. Now, you can see actually that, first of all, this network is actually pretty much stable across the year. So if I plot this dispersal network, I always observe the same thing. So for instance, this means that Swamp Lake is actually a source of pollution, and that mercury is actually moving from Swamp Lake to all the other. But secondly, you can see that overall, you have an increase in mercury concentration for any water body that we consider, okay? And again, this trend is actually starting probably much earlier than 1995, and we are trying to link this trend to actually the variation of fish biomass that we observe system-wide. So the bottom line, I can repeat this analysis for many other areas and many other species. The bottom line is that, first of all, in my opinion, it's really important to analyze the data and work with the people in the field and trying to guide actually that field monitoring using mathematical models. And the second thing is that mathematical models can actually help in finding out pathways that at the current state or in the future state may actually affect both the animal population and the human population. So my overall goal to synthesize in this project is really to try to discretize all the physical phenomena that actually occur on the landscape, to try to find the sources of the problem at the current moment or in the future. And I believe that public health is in need of this tool, as well as the veterinary medicine community, but also the ecological community that is actually affected by this issue. So yeah, I think I can open up to questions if there is any.
Yeah, thank you, Matteo. That was wonderful. It's so neat to hear about this research happening, you know, that so directly affects the community.
Right.
And I appreciated that you mentioned that understanding this kind of bioaccumulation and the source of these pollutants could help to direct the fish advisory communication to help people be safer and to, you know, be able to fish, but not get sick based on the species or the time. But I had a question.
Sure.
Do you see opportunities for the community of Grand Portage or other communities dealing with similar issues to build on or contribute or utilize your research to address these issues?
I mean,
what action can the people who aren't using these mathematical models, how can they utilize those to create a more sustainable community?
Well, I think there should be a distinction between what people are going to do and what we are doing. No, the people, the local stakeholders in the Grand Portage Reservation, they're certainly not using the mathematical model. I'm using the mathematical model, and I am trying to work with them and the other collaborators in trying to understand, first of all, the sources of the issue, trying to understand what is actually causing the presence of the micropollutants, trying to decrease the level of micropollutants. And we are also thinking to release this food consumption advisory that are both dependent on time and dependent on space. So obviously there is one part that is more related to environmental management, and that is done by the people in Grand Portage. And they have, again, this environmental lab that is quite efficient. So I work personally with this guy that are actually alumni of the University of Minnesota, I think in the ecology program. And they collect data, they get me data back, I analyze them. And so it's like a team approach. Not everybody needs to be able to run a mathematical model, and not everybody needs to be able to go into the field. It's more about how we put our forces together to generate something that is useful for the whole population. That's how I see it. And I think that it has been really nice so far. Because the collaboration is very strong, and it's not always the case to find a team that works so nicely together, just because somehow the disciplinary boundaries create some obstacles, and there is this idea that somehow mathematical models need to be accessible to everybody, which I don't think that's the case. I think the output should be translated to the local population for expected use of the findings.
That's wonderful. Yeah, it seems like a really great example of a collaboration that works well to come out with a really useful end result. How long is this project going for? Are you pretty much wrapped up with your data collection and analysis, or are you still continuing?
No. We started last summer with the preliminary funding from the Institute of the Environment, and we got actually some preliminary funding from the M-Drive program. I don't remember precisely that funding source. And the funding, the official funding from the LCCMR starting next August, and so we're going to continue for 2 years. So we're going to have more than 65 sites that are sampling sites in which we are sampling water, soil, subsistence species. Most of them are fishes, and that's how it's going to be the situation. We didn't sign yet the sampling frequency, but that's going to be related to the hydrological cycle, essentially. So we're going to sample in the winter, in the summer. And again, don't ask me too much because I'm not really a field person, but I'm trying my best to try to indicate what are the best location and the period for doing that field monitoring.
Awesome, thank you.
Yeah.
That is super great. I'm excited to share this with the work groups around our region to kind of talk about what you said about collaboration and making use of everyone's talent. So it was great. Yeah, we're going to transition now into the second part of our webinar, where Molly and I will share a little bit about our upcoming SCC meeting. But if you have questions for Matteo, please continue to put those in the chat box, and we can kind of revisit that after this next section. So let's see, Molly, do you want to start with a little background about the Statewide Coordinating Committee meeting?
Yeah, I'd be happy to. Can you guys hear me okay?
Yep, sounds great.
Okay, good deal. So yeah, thank you, Rose. Again, just wanted to thank you to Matteo for that really terrific presentation. That's a fascinating study, and I'm looking forward to reading and learning more about it. But as Rose mentioned, we'll be spending a little bit of time today highlighting our upcoming Statewide Coordinating Committee meeting that you're all invited to attend on November 3rd for our deep dive discussion into natural resources. And also too, just to mention, I'm Molly Zins. I staff our Central Sustainability Partnership, which is one of our 5 regional partnerships serving all of Greater Minnesota. So I'll just start with briefly touching on a little of the big picture background of the partnerships to give a bit of context into how this leads to our statewide coordinating committee format and convenings, and really the importance of engaging all of our citizen partners in these discussions. And I'll just mention briefly too, our statewide coordinating committee is often referred to with the short acronym of SCC. And so in the interest of brevity, I'll probably call it SCC moving forward for the next few minutes. But our 3 bedrock principles of the partnerships really drive all of our work. And this, of course, includes our FCC convenings. Our principles start with the priority to develop vibrant partnerships between the citizens of Minnesota and our land-grant university. Also fostering sustainable development in our 4 focus areas. And finally, promoting active citizen leadership across Greater Minnesota. And many of our SCC members and participants include our regional board members from across our 5 regions. We have governing boards in each of the 5 regions, and for those who aren't familiar with this, this entity of the partnerships, the governing boards serve as the decision-making bodies in allocating all of our project funding and resources and really in guiding our programming and our priorities. Our SCC also includes many of our citizen advisory work group members, and we know many of our webinar viewers in these webinars are serving both on a work group and or a board. But for those of you who might be watching now or on the recording, and if you don't have familiarity with this part of our work, please let us know if you're interested in learning more, and we'd be happy to share with you opportunities as to how you could engage in these roles. So we use our SCC meetings to have deep dive conversations into our 4 focus areas, rotating the focus topic throughout the year. And as I mentioned, we're having our natural resources deep dive this November 3rd. And I'm happy to share that we can trace the formation of this very webinar series and all of Rose Clark's really excellent work as our Conservation Corps member back to one of our earlier SCC meetings from a couple years back. At that meeting, in that natural resources deep dive discussion, a group of folks representing all 5 of our regions and including regional board members and work group members identified several key needs for our natural resources work as the partnerships, including the need to strengthen our natural resources in-reach, as we call it, into the University of Minnesota. So in other words, strengthening the way we work that helps our boards and our work groups, our project partners, connect more directly with University of Minnesota faculty and students working in this field. We also identified the need to facilitate more connections across the 5 regions, across the 5 regional boards and regional work groups, including identifying common priorities and resources, and seeking opportunities to better leverage what's already happening on the ground in our natural resources work across the regions and what could be happening in some of our regions. Also, we sought to better communicate the partnership's natural resources priorities, our portfolio of natural resources work, and ultimately the impacts of both individual projects as well as collectively where we're moving with our natural resources body of work. And finally, we really identified the need to offer more opportunities for work group members, board members, project partners, other citizen collaborators, and university partners to engage with us in a variety of different formats around the natural resources discussions and priorities that our citizen partners are helping us lead. So as SCC participants further refined these priorities, we ultimately sought and secured the funding to create Rose's Conservation Corps position, and we're now fortunate enough, as you've heard, to really have her on our team and coordinating all of this great work. And so I just wanted to share that as one example of a direct result of an SCC discussion that started as just a round the table discussion a couple of years ago, engaging a number of our citizen partners. And with that, we invite you all to join us at upcoming SCC meetings to help craft future priorities and strategies in our work. And with that, I'll turn it over to Rose, unless, Rose, maybe I didn't touch on something that you asked me to. So please just let me know if I left something off that I should cover.
No, it was a wonderful overview. Thank you, Molly. Yeah, one of my other favorite SCC success stories from before I joined in January was at I think it was a CERT, so the Clean Energy Resource Teams collaborated with, folks at one of these meetings and came up with a plan for a low-watt light bulb sales fundraiser for youth groups to make an influence in their community while they're raising funds. So just really awesome ideas come out of these meetings because everyone is together and sharing ideas, and good things happen. And we're lucky enough to have the focus be on natural resources in 2 weeks on November 3rd, the Thursday, from 9 a.m. to 4 p.m. And I wanted to give a little bit of an overview of the schedule for the day. for those of you who are interested, but that might be interested in hearing more about the specifics. So we are very excited to be welcoming the Council of Deans and President Kaler of the university to help us kick off our meeting on that Thursday, November 3rd. We'll go through some business and then hear some regional and statewide reports with a really focus on the natural resource aspect. We'll have a nice lunch, and then we're having a Grand Challenges panel. So for those of you who aren't familiar, the University of Minnesota has identified 5 Grand Challenges in a variety of different focus areas. One that relates really well to our natural resource work groups is that of clean water and sustainable ecosystems. So addressing all the different contributing factors to achieve that Grand Challenge. We've invited 3 panel presenters to come and share with us their perspective on what the emerging issues are in natural resources and give us some insight into the work that they do. So our 3 presenters are Shannon Kesner. She's a wetland specialist with the Fond du Lac Office of Water Protection. Fond du Lac Nation is doing really great work in natural resources, just really innovative things, and they actually just recently completed a climate adaptation kind of analysis and plan forward for their natural resources. So we're excited to hear from her about that. May Davenport is the director of the Center for Changing Landscapes and also the Department of Forest Resources. She's going to be sharing with us, her perspective. She's done a lot of work with communities and watersheds. And then our 3rd panel presenter is Nate Meyer. He's with the U of M Extension over in Cloquet, program leader for the Fish, Wildlife, and Conservation Education. And he's been working with Molly and some others on climate change and climate adaptation training and has really great insight himself into natural resources and how shifts in our climate might affect communities. After our panel, we'll have some action-oriented breakout sessions, and these will be times for people to collaborate with people from other regions around different topics, breakout groups. Some of those are more operational, more process-oriented, like, for example, what does an effective work group look like? And that can be in any of the focus areas, but specifically, what does that look like in natural resources? How can we be most effective in our role as a community university connector? Other breakouts might be more topic-focused on specific issues. Some things that have come up in the regions that people seem to have a lot of questions about how to approach include the buffer implementation and how that affects communities and how that affects the ecosystem, or things like watersheds or soil carbon. It could be anything. So we'll have a few topics to choose from, but we also will invite people to come up with topics that they'd like to further discuss based on the information shared earlier in the day. At the end of the day, we'll have some time to share what we came up with in our breakout, and then we'll adjourn around 4. The purpose of this day, of this FCC meeting, as far as the natural resource focus area goes, is we really want to know how do we strengthen our work in natural resources, and how do we address emerging issues in this topic. I'm hoping to get direction from the members of the region and the members of the FCC committee to really craft my work plan for this coming year as I start my 2nd year in the Conservation Corps position where I am lucky enough to find myself. Yeah, so that's what this Thursday, November 3rd meeting looks like. Again, we would welcome any of the RSDP members that want to join us on the Minneapolis campus. And Matteo, we'd also be super happy to hear from you if you're around that day as well. One more little invite to send out to folks is that the night before this FCC meeting on Wednesday, November 2nd, from 4:30 to 7 p.m. on the St. Paul campus, we'll be having a Institute on the Environment, ION-E reception. They are a wonderful unit of the university. We heard from Jessica Hellman in an earlier webinar about all of the really great work that ION-E does to support students and staff people who are making great strides in sustainability all throughout the university system. Matteo's work was partly sponsored or funded, I think, through ION-E as well. And we'll be playing the Watershed game, which I'm really excited about. It's an interactive educational tool. It's facilitated by members of the U of M Extension Water Resources team. There in the photo on the slide, you can see our Southwest work group member, Karen Perry, leading a game. We'll be leading 2 games to accommodate the folks that we have. And yeah, you can learn more about it at the link, northlandnemo.org/watershedgame. I am quite excited to be playing that. So that's on Wednesday the 2nd, the FCC meeting on Thursday the 3rd. It'll be a really great time, and hopefully we'll get a lot of good discussion out of it. Did you have anything else to add, Molly?
No.
If anyone has any questions, you can feel free to enter them in the chat box as well. Sorry, Molly, did you have anything else to add?
Oh, no problem. I was just unmuting. Nope, I think you covered it well. Thank you.
Yeah, sweet. Well, with that, I'm going to turn on the poll real quick. I know we only had one official presenter instead of our usual 2, but if you wouldn't mind, for those of you who are attending, answering this poll about how much you learned and what content was most interesting or useful to you, that would be really helpful. And, yeah, Matteo, as someone fairly new to the partnership, do you have any questions about us or about the meeting? Might be muted. Yeah, I'm really looking forward to the day. I really have a lot of fun at these meetings. It's neat to see all the folks, and I did want to point out too that in this picture of, in this slide with all the pictures of the statewide coordinating committee meetings, I really love this one from this July. We always have one meeting a year, has been the trend in the past, of going to visit a region in greater Minnesota. So this July we were in International Falls in the Northeast region, and kind of a focus on the Northwest region projects, and it was just a really great day and great trip. Okay. Looks like we're getting some responses in. So with that, I think we'll wrap up. Thank you again to Matteo for sharing your research and your perspective. I hope that we can find more ways to utilize research like yours and work with you. And thanks to Molly for giving that awesome insight into the FCC meetings. If anyone has any further questions, go ahead and give me an email. My email address is rmclarke, C-L-A-R-K-E, @umn.edu. And I hope to see lots of board and work group members at the FCC meeting to talk about natural resources. So have a good rest of your day.